Everybody's ready? So today I'm going to say about just a few words about the last of the things with lipid metabolism, and then I'll start talking about photosynthesis. And I'll talk about photosynthesis probably for today and part of Wednesday, and then we'll finish everything up with nitrogen metabolism on Wednesday and Friday. So as I said earlier, I will be available after class today if you want to come and talk to me about corrections and so forth for the exam. Those things are all due by 5 p.m. tomorrow. So I know I've been a little hard to get a hold of, but I'm trying to accommodate you as best I can. And if you're still having difficulty connecting with me, let me know. But I think hopefully we should be able to get everything done by today, or by tomorrow, actually. So, but hopefully tonight will help a lot. Okay, so last time I talked about receptor-mediated endocytosis, and I said that this was the way in which cholesterol actually makes it into cells. So when we think about dietary cholesterol has traveled this interesting path. It has gone from the intestine, it's gone into chylomicrons, it's gone back to the liver after those chylomicrons shrink. And by the way, those shrinking chylomicrons are called chylomicron remnants, because that's what they are. They're remnants of their former self. Excuse me. And they've gone back to the liver. They've gotten incorporated into the liver. And then the liver has said, oh, the cells or the body is needing some of this, so it's packaged them up as part of the VLDLs. The VLDLs were made into the IDLs. The IDLs were made into the LDLs. And finally, the cholesterol is internalized into a cell through this process of receptor-mediated endocytosis, and that's what you see on the screen. Now, I want you to remember, of course, that fats and other lipids are traveling in those lipoprotein complexes as well. So it's not just cholesterol, but this is the way that dietary cholesterol makes it directly into cells. So it's a pretty convoluted path, and it means that along that path, dietary cholesterol is traveling through the bloodstream a pretty fair amount. Okay? It's traveling through the bloodstream a pretty fair amount. So I'm going to say a little bit about that in just a couple of minutes. And I don't have any particular thing here to show you other than the fact that these LDLs, which are up here, are being internalized through this process, and it's this process that we call receptor-mediated endocytosis. Once these things get inside, all the goodies get unpackaged, the cholesterol gets gobbled up and then used by the cell, and the other components then get recycled as necessary or appropriate. Mariah?
When they get endocytosis with that, are they still remnants, cholesterol remnants?
So remember, these are LDLs. These are not cholesterol remnants.
Okay, so they get like...
So the cholesterol remnants got taken up by the liver,
Right.
okay, and then they were repackaged as VLDLs, which became IDLs, which became LDLs. So they're not remnants at all, no. Once they're taken up by the liver, they're no longer remnants. Okay, so that's what receptor-mediated endocytosis is about. And you can see there's other things that are coming in by this mechanism. Fats can come in this way, okay? Fat-soluble vitamins can come in this way as well. Okay. Now, the last thing I wanted to say, one of the complexes I haven't talked about were the HDLs, the high-density lipoprotein complexes. So I want to say a word about those. And I want to say a word about another word about LDLs as well. So if you go to your doctor and the doctor examines you for cholesterol levels, you will commonly hear the doctor or somebody associated with him describe what's called good cholesterol and what's called bad cholesterol. Okay? So the bad cholesterol is measured by the level of LDLs that are found in your bloodstream. And the reason that it's called bad cholesterol is because it's this form of the lipoprotein complex that's associated with atherosclerosis, that is, clogging of the arteries, blocking of the arteries, and so forth. So LDLs are associated with atherosclerosis. It's the LDLs that actually have the highest concentration of cholesterol, because remember, all the other goodies are being taken away and the cholesterol is just sitting there. The fats get dissolved away by lipases and so forth. So the cholesterol as a percentage of the total is actually increasing in this process. So why is it bad cholesterol? Well, it turns out that the LDLs are very susceptible to reactive oxygen species. Now, reactive oxygen species I've briefly mentioned before, and I'll remind you that they are a form of oxygen that is very reactive. You remember superoxide dismutase helped to break down reactive oxygen species. And reactive oxygen species are produced by many different mechanisms. Reactive oxygen species are something we would think of as harmful. They're harmful because they react without control and indiscriminately. Reactive oxygen species can give rise to mutation. They can give rise to damaged proteins, etc. And one of the things that they can do is that they can react with LDLs. So when they react with LDLs, the LDLs can become attached to arteries. They can become attached to arteries. And that's the first step in the formation of a blockage known as an atherosclerotic plaque. A blockage known as atherosclerotic plaque. A plaque is a block. A plaque is a P-L-A-Q-U-E, by the way. Okay? P-L-A-Q-U-E. That is a blockage of some sort. And this happens not because of one LDL being stuck to an artery, but because there may be a cluster of them, and that cluster may arise as a result of, first of all, a lot of reactive oxygen species. And if that should happen, then the immune system actually sees it as a foreign body and attacks it. The macrophages will attack the LDLs. The macrophages are very important in the immune system, and they can really glom onto something because their aim is in destroying invaders. Well, if you have a bunch of LDLs stuck in a particular place, those macrophages, when they glom onto there, it looks like a big foamy mess because the cholesterol all spills out, the macrophages are going crazy, and they're called foam cells because they're just full of this junk. Okay? As that happens, okay, hardening can occur, solidification can occur, and that's how that atherosclerotic plaque actually forms. So that's why LDLs are known as bad cholesterol, because they're favoring the formation of that atherosclerotic plaque. Okay? And where do HDLs fit into this? HDLs actually help reduce the concentration of damaged LDLs in the artery system. They actually start salvaging some of these pieces and taking them back to the liver so they don't cause problems. So when your doctor looks at your profile of lipoprotein complexes, the doctor will be happier the higher your HDL levels are and the lower your LDL levels are. And he or she will be unhappier the higher your LDLs are and the lower your HDLs are. Okay? So you want to have high levels of HDLs, low levels of LDLs. Does that make sense? Questions about that? Okay. Well, let's move on to talk about photosynthesis. So photosynthesis is responsible for the harvest of energy on the planet Earth. Plant cells and photosynthetic bacteria are the solar cells of our planet. When we think about solar cells, we think about capturing sunlight and making energy from it. Okay? Plants and photosynthetic bacteria capture sunlight and make glucose from it. And glucose, of course, can be used as an energy source for everything that the cell does. Okay? Now, let's think about this overall, or let's learn a little bit about this overall process. So I'm only going to focus on plants. I'm not going to talk about photosynthetic bacteria, some of the processes occurring here occur in photosynthetic bacteria, but photosynthetic bacteria are somewhat more complicated because they don't have chloroplasts. They have it sort of mixed up with all the other things of their membranes, and it gets a little complicated to talk about. So I'm just going to talk about plants in this scheme of things. We know, of course, in plants that plants have organelles known as chloroplasts, and it's in the chloroplasts where the chlorophyll is located. Now, chlorophyll is a pigment. Chlorophyll we associate with green, right? And chlorophyll is the part of the complex that absorbs light energy and allows the cell to do something with it. So when we think about this process of photosynthesis, the light energy is grabbed by the chlorophyll. Okay? Now, we're seeing a cross-section here of a chloroplast. And when we talk about the chloroplast, we're going to focus on a couple of things within it. The chloroplast is actually a little bit more complicated than the mitochondrion is in our cells. By the way, plants have both mitochondria and chloroplasts. It's not an either-or. They have both mitochondria and chloroplasts. But the chloroplast, you can see, has some complexity to it. The chloroplast, like the mitochondrion, also has DNA of its own. The chloroplast and the mitochondrion are the only two organelles that have their own DNA. The chloroplast DNA is bigger than the mitochondrial DNA. It codes for more genes than the mitochondrial DNA does. Now, that's not shown in this figure at all, okay? And we're not going to focus on it all. But I want you to be aware of the fact that chloroplasts do have their own DNA. It's also a circular DNA. Okay, so I said we're going to focus on a couple of things, and the things that we're going to focus on are really right here and right out here. Okay? Right here, we see something called thylakoids or thylakoid space. So these structures here are called thylakoids. These flat layered things are called thylakoids. The thylakoids have two components of interest to us, and we'll draw them as a parallel to the mitochondrial membranes. For example, there's an inner portion, okay, that's called the thylakoid space. We will think about that in the same way we think of the mitochondrial matrix. Okay? We have a thylakoid membrane, and we're going to think about that in comparison to the inner mitochondrial membrane. And last, we have a stroma. And stroma is just the space out surrounding there, and that'll become important as I talk about that later. So the stroma contains fluid, okay, and that fluid bathes all of these structures that are in here. So those are the components, the thylakoids, the thylakoid space, the thylakoid membrane, and the stroma. Okay. What's happening in the process of photosynthesis, you probably have learned from other biology classes, is that photosynthesis occurs in what we call two phases, a light phase and a dark phase. Okay? Now, they're really misnamed. Okay? The light phase, of course, requires light. The reactions of the light phase can't occur unless there is light. The dark phase, however, does not require dark. It simply does not require light. Okay? So the dark phase and the light phase. All right? In the light phase, something very important happens as one of the first steps in the process. And the very important thing that happens in the light phase is the production of oxygen. We know, for example, that in primitive Earth, before plants and photosynthesis evolved, we know the composition of the Earth's atmosphere. And there was virtually no oxygen in the Earth's atmosphere. The oxygen that's in the atmosphere today, okay, arose largely because of photosynthesis, releasing oxygen. So the release of oxygen by plants was essential for early Earth, okay? And it's essential for later Earth because we need oxygen today to breathe for our electron transport system. So that's a very critical thing in the light phase of photosynthesis. In the dark phase of photosynthesis, carbon dioxide is taken out of the atmosphere and it's built into sugar. Specifically, it's built into glucose. Okay? Now, when we think about global carbon dioxide levels increasing, as is happening with climate change and as is a driving force for climate change, okay, one of the mitigating factors for that is the use of carbon dioxide by plants, because plants take carbon dioxide out of the atmosphere, okay, and thus reduce levels of carbon dioxide in the atmosphere. Now, we're dumping more carbon dioxide into the atmosphere way faster than plants can take it out, but it's not as bad as it would be if plants were not taking it out. We would like to get plants to take more carbon dioxide out of the atmosphere. Okay, so we have a light phase and we have a dark phase. There's the structure of chlorophyll. You've seen a structure very much like that before. What structure did you see that was like that before? Heme. Okay, so heme has almost the identical structure, one difference being it has magnesium in the center instead of having iron in the center. Like heme, it has that flat ring, and we see these long attached things out here that actually embed this chlorophyll in membranes. Okay, so this guy is embedded in membranes because of this long side chain. Okay, now, what I want to do here is talk about the molecular basis of photosynthesis. And I'm going to start by describing what people call the Z scheme. So I'm starting with the light phase of photosynthesis. And we're going to see that the light phase of photosynthesis occurs through two photosystems. Two photosystems. What's a photosystem? As we're going to see, a photosystem is the equivalent of our electron transport chain. Now, instead of having one, they have two. Plants have two, okay? And we're going to talk about that. But in the photosystems, what's happening is electrons are going to move and protons are going to be pumped, all right? Just like we saw in electron transport, except for there's an interesting wrinkle, and I'll talk about that as we get to it. So let's look at the Z scheme of photosynthesis where we see the process occurring. Now, one of the things I'm going to spare you in photosynthesis is requiring you to name all of the individual components, all right? That's a lot of stuff. And you can see there's a lot of different proteins and other molecules that are involved in the transport of their electrons. The process starts in something called photosystem 2. And it's called that because it was found second, not first. Transported back to photosystem 1, and then we see all these arrows are indicating movement of electrons. Movement of electrons. So let's start over here on the left side with the process with the absorption of a photon. Okay? So a photon of light hits and causes this whole process to get started. All right? So what we have over here, the process starts not here, but it starts here in the photosystem. And the photosystem we're going to see is going to pull electrons away from water. Now, this is a complicated thing. What plants are doing is they're actually oxidizing water. Water is losing electrons and protons to become oxygen. Now that takes energy. You've got to do a fair amount of energy to pull those electrons and protons away from water to make oxygen. But we're not starting here. We start here, and you'll see why in a second. Okay? So here's this photosystem here. Okay? The two photosystems are distinct based on the wavelengths of light that they prefer to absorb at. So there's 680 means it prefers to absorb light at 680 nanometers of wavelength. Okay? I don't care if you know that. I care if you know photosystem 2 and photosystem 1. Okay? Now, a photon of light hits photosystem 2. When a photon of light hits photosystem 2, this photosystem has that chlorophyll sitting there, and that chlorophyll has an electron that can be excited. Hey, Ahern, they can be excited. It gets raised to a higher energy level. That's what happens in this process. So an electron in that photosystem gets raised to a higher energy level. Okay? It's excited. When you get excited, you jump up and down, right? Well, maybe you don't. Maybe I do. Okay? I jump up and down when I get excited. When I get really excited, I do jump up and down. And that you can imagine that electron jumping up when it gets excited. It's got higher energy. It goes to a higher level. Okay. This electron leaves this base system right here. It leaves it. Well, if an electron leaves, what does it leave behind? It leaves behind a hole. And what wants to fill that hole? Another electron. Well, this electron is getting ready to go through this whole scheme. It's actually a pair of electrons, but this pair of electrons is getting to go through this whole scheme. There's nothing that's going to bring it back. Nothing's going to bring it back. When I start jumping up and down, nothing calms my excitement. I'm ready to go and celebrate the excitement that I have. And these electrons are ready to go this way. Now there's a hole of 2 electrons right here. Well, that's a big hole. Okay? Electrons, as you know, are reactive. In absence of electrons, make this reactive as well. So what happens is this guy right here pulls 2 electrons from this oxygen evolving complex. And what does this oxygen evolving complex contain? It contains manganese. Not magnesium, manganese. Chemical symbol Mn. Okay? Well, now manganese system is lacking 2 electrons. And where in the world can the manganese complex get 2 electrons? Water, because water is surrounding everything there. So the manganese complex is actually responsible for taking those electrons from water. And when it takes electrons from water, water loses its protons at the same time. And that leaves behind oxygen. So the very first things that are happening, the excitement of electrons, the robbing of electrons again yields oxygen. Now I'll slow down and stop there and see if you have any questions. Yes?
Why doesn't the photosystem 2 just pull it from the water to fill it?
Why doesn't it pull it from the water? It's not energetically favorable for it to do it. It's much easier for it to leave a hole, and the hole actually is pulling it from the water indirectly. Right? So it's the creation of that hole that's critical. In other words, to do it from photosystem 2, light would have to be exciting electrons from water to make that happen, and that's not energetically favorable. Yes?
So the electron is exciting and leaves the second electron.
Both. I said one, but it's actually 2 electrons that are leaving. Yes, it's a pair. Yes?
What exactly was the manganese complex? Is that part of the photosystem?
The manganese complex is right here. Okay, so it's not a part of photosystem 2, but you can see it linked to it.
So it's the oxygen evolving complex.
It is the oxygen evolving complex right there. It's due to the manganese-containing complex that oxygen is produced. Other questions? Okay. Well, I haven't accounted for the electrons yet. All right? All I've said so far is that we've excited this guy. The hole got replaced by this, and so we've made oxygen. That's what we've done so far. Now what I want to do is follow these electrons that have been excited through the system. Okay? We're going to follow those electrons through the system. Well, here's these excited electrons. They want to go out and party, right? And so what they do is they pass through a series of electron transporters that are embedded in that thylakoid membrane. These electron transporters are embedded in that thylakoid membrane. They grab an electron, they pass it on. They grab an electron, they pass it on. They grab an electron, they pass it on. Okay? And ultimately, those electrons end up in photosystem 1, which is another way for light to affect this system. So we started in photosystem 2. We've now gone to photosystem 1. And in the movement of these electrons, here's the wrinkle I told you about. As the electrons are moving through this system, protons get pumped. That's not the wrinkle. The wrinkle is the direction that they get pumped. In the case of the mitochondrion, I'm sorry, not electrons pumped. Protons get pumped. Protons are pumped, not electrons. Now I'm starting to confuse them myself. Protons are pumped as a result of the movement of electrons through these complexes. Okay? The proton pumping that happens in mitochondria pumps the protons out of the mitochondrion into the intermembrane space, right? In the thylakoids, protons are being pumped into the thylakoid space. They're taking it from the stroma and they're pumping it into the thylakoid space. So instead of going out of something, they're going into something. And it turns out that's not a big deal. It's not a big deal at all, because it's just another way of making a gradient. Instead of having a higher proton concentration out than in, these things have a higher proton concentration in than out. And the protons now, instead of wanting to get in, want to get out of the thylakoid space. Everybody follow that? And here's the thing you're going to like. The ATP synthase that the protons travel through is almost identical to complex 5 that we see in the mitochondrion. There's a rotor, there's a turbine that's turning. And that turning of the turbine is what's responsible for making the ATP. Really cool, okay? So again, we see a proton gradient is responsible for making ATP. This is the third type of phosphorylation that I talked about earlier in the class. Okay? I talked about substrate level phosphorylation, which involved a high energy intermediate like PEP. I talked about oxidative phosphorylation, which is what occurred in mitochondria. And now I'm talking about photophosphorylation, and we call it that because light is involved in the process. Photophosphorylation. Again, like the oxidative phosphorylation, we see a complex, an ATP synthase, that is spinning a turbine, and ATP is being produced. All right, we haven't gotten to to photosystem 1 yet. We're now at photosystem 1. To complete this process, another photon of light has got to come in, and it excites these electrons here. This photosystem prefers light at a wavelength of about 700 nanometers. The electrons get excited, just like they got excited before. Okay? And now they move through this system over here. So there's more complexes that they're going through. They're again in the thylakoid membrane. They're membrane bound. Okay? The one thing I want you to know in terms of names is this guy right here. the electrons end up at a protein called ferredoxin. Ferredoxin. Ferredoxin is the final place where the electrons end up before they reduce NADP. So when the electrons reduce NADP at ferredoxin, okay, and by the way, it's called NADP reductase, the enzyme that does this, we end up with, or the plant ends up with, NADPH. The enzyme does the final reduction is NADP reductase. Is that what you're asking? I interrupted what you were saying. I'm sorry. Yeah. I thought I was reading your mind. So, yes, sir. Ferredoxin is the last protein that's holding on to electrons. So the electrons from ferredoxin are donated to NADP to make NADPH, and the enzyme that catalyzes that is NADP reductase. Yes, sir. Say it again, I'm sorry.
In photosystem 1, when those electrons get excited again, what fills that hole?
What fills that hole? Well, it turned out these electrons were actually extra because they just ended up here, right? So we're equalizing everything out, okay? Yes?
So for photosystem 1, it can't go through that step without photosynthesis.
That's right.
So, but what you're saying with him, there's already electrons there, so
These electrons aren't problematic. There's not a hole as such that's there. There's not a hole. So this will all be fine here. Because remember, this is going continuously. So we can think of this going like a circle, right? So I started you here, but in fact, this is just going on and on and on, just like any other cyclic process. Okay, well, now let's think about this. This really turns oxidative phosphorylation on its head. It turns it upside down. Let's think about it. In oxidative phosphorylation, what was the electron source? NADH or FADH2, right? In this process, what's the electron source? Water, right? In oxidative phosphorylation, what's the ultimate recipient of the electrons? Oxygen. And what's the product? Water, right? And here, what's the acceptor of the electrons? NADP. And what's the product? NADPH. So in this process, we're making an electron carrier, and we're starting with water. In oxidative phosphorylation, we're starting with an electron carrier, and we're ending up with water, right? In a lot of ways, we're just going backwards. Now, of course, that's schematically we're going backwards. The steps are different in the process. But you see that basically, if we look at only the starting and end points, they're just opposite processes. But the beauty is they both produce ATP. And you might say, how is that possible? How is that possible that a system, in the case of oxidative phosphorylation, that's starting with electron carriers and ends up with oxygen, or ends up with water, produces ATP. But if I start with an electron carrier and I end up with water here, I still produce ATP. How is that possible? That sounds like a cycle that just goes on forever, and that's not possible, right? Well, it is possible, and it's possible when you put in energy. Futile cycles work when we put in energy, right? We don't want futile cycles because we waste energy. They go around and around, they produce heat, and they don't do anything good. Here, the energy that drives this is photons. Photons are what make this energetically unfavorable process favorable. This is a plot of energy. Look at this. The energy of this guy is higher than the energy of this guy. But it ends up working because the photons of light provide the driving force to make the overall thing happen. So photons drive the process, provide the energy necessary for this to happen. Now, the products of this are two, or actually three if we want to think about it. Here's a product, oxygen. No energy component there, but it's pretty important for all the rest of us out here who breathe it. Here's a product, ATP, produced by photophosphorylation. Here's a product, a reduced electron carrier. This and this will be essential in the dark phase of photosynthesis. They'll be essential for the dark phase because they are energy sources, and in the dark phase of photosynthesis, we need, or the plants need energy because they're going to pull carbon dioxide out of the atmosphere and they're going to build it into glucose. That takes energy, just like gluconeogenesis took energy in us. And we weren't even taking carbon dioxide out of the atmosphere. Andrew. Okay, good question. So Andrew's question is ATP produced by the chain or by the chemical gradient generated by the chain? It's produced by the chemical gradient generated by the chain. It's the proton gradient that comes from this movement that causes ATP to be produced. No, it's made right here in the light phase. One for each one of these. Okay? Yes?
In oxidative phosphorylation, ADP was like the regulator. Does that still apply here?
Okay. Oh, this is a very good question from Anna. So in oxidative phosphorylation, I said ADP was limiting. Are there any limits here? Okay. What do you suppose? Well, light would be a limit, okay, but let's say we've got plenty of light. It turns out that the reason her question is good is that it turns out that there is something that's limiting here. What do you suppose it would be? It's not water, because the plants generally have plenty of water. Not the orientation electron. Andrew? NADP. This thing on a bright sunny day becomes limiting. Okay? It becomes limiting. And plants have evolved an interesting sidestep around this, where they don't require NADP. And I'll show you that probably tomorrow. Okay? But NADP does become limiting for these plants. Okay. Well, let's see. Where was I? Here's another view of the same thing. And this is drawn to show you the same thing, looking kind of like what we see in oxidative phosphorylation in the mitochondria. Okay? Here's that manganese complex. Here's where the light is coming in, pulling out these electrons, and the manganese complex makes up the deficit, and then water makes up the deficit inside of there. Okay? Here's the movement of electrons in blue. And you can see it's a more complicated movement than was happening in electron transport in our mitochondria. We have two big photosystems. We have a complex here where we see protons being pumped as part of that system. And I haven't recorded any names here. Okay? You can see these small molecules that are carrying, this is plastoquinone, plastocyanin. I'm not going to make you know those. But plastoquinone and plastocyanin are kind of like the coenzyme Q that we talked about in the electron transport system, small and able to move, and it's moving between bigger complexes that don't move so well. Okay? Ultimately, there's our ferredoxin that's getting those electrons, and there's the attached to here, the ferredoxin, is the NADP reductase that's adding the protons and electrons to make NADPH. Over here is what's happening with the proton gradient. The protons are being pumped in, causing a high concentration of protons on the inside. Those protons want out, and how do they go out? Through the ATP synthase. And it's almost identical to the one in the mitochondria. Pretty cool system. Without this, life on Earth doesn't exist as we know it. It doesn't exist as we know it. We don't have oxygen if we don't have this system functioning. You're not responsible for this, but here's one of the small molecules that carries those electrons. And you probably don't remember, but coenzyme Q didn't look very different from this. It had a long tail, and it had the ability to carry electrons in a ring. And that's what plastoquinone and plastocyanin both have the ability to do. Okay. There actually is coenzyme Q, ubiquinone. Okay. So you can see they're very similar in the way that they carry electrons. Okay. Now, let's see, I don't want to talk about that yet. This I just throw at you to show you again what's happening in this overall process. It looks a lot more complicated. That's why I don't start with this. And again, there's no detail in here that you're going to need to know beyond what I showed you in that earlier figure. Okay? But again, it's showing a little bit more detail, a little bit more of the complexity of what's happening. This is, of course, the thylakoid membrane. Here's the thylakoid space. Here's the stroma. Okay? So you can orient yourself with respect to all of that. And I wasn't going to talk about it, but I think I will here since Anna has asked the question, and that has to do with photophosphorylation. Okay. Photophosphorylation is important for plant cells because when they're in bright sunlight for a long period of time, they get real charged up like a battery. They get real charged up like a battery. Okay? And yes, they can produce a lot of ATP, and you might think ADP is a limitation, and at some level it might be a limitation. But the bigger limitation for plants in bright sunlight is actually NADP. NADP. Because that's the ultimate electron acceptor. And if plants don't have something to do with those electrons that get excited, then this cycle of pumping protons cannot work. So let's imagine that, as I've told you, NADP is more limiting than ADP is. So cells want to make as much ATP as they can. But if they run out of NADP, they can't make as much ATP as they have ADP. So the ADP just sits there doing nothing. So rather than have NADP limit the amount of ATP that a cell can make, they've evolved this system called photophosphorylation. Cyclic phosphorylation. Okay? Now what I'm going to show you here is similar to what occurs in photosynthetic bacteria. Their mechanism is similar. Okay? It involves photosystem 1. It doesn't involve photosystem 2. In photosystem 1, we see excitation of electrons, but instead of passing those electrons through a chain that leads to ferredoxin, they leak back to this complex before there where the proton gradient was. So let me go back and show you. Okay. So normally in the let's go to the Z cycle. I'll show you the Z scheme better. In the Z scheme, we start photosystem 2. We go through this proton pumping complex. We go to photosystem 1, and we go to ferredoxin. In cyclic photophosphorylation, we're going to see excitement starting here. And then instead of going this direction, it's going to dump electrons back this direction. So it's going to dump the electrons back into this thing. This whole process over here will not occur. There won't be any oxygen generation. We're only going to see this going on like this. So in photophosphorylation, I keep saying photophosphorylation, in cyclic photophosphorylation, let's see what's happening. Okay? There's complex 1, exciting electrons, okay, from the photon coming in. Electrons leak back into here. Well, this thing doesn't care where the electrons come from. It just cares that electrons move through it. And when electrons move through it, what's happening is protons are getting pumped. Okay? The electrons move through it and ultimately end up in bounding to oxygen to make water, very similar to what we saw in the mitochondria. But more importantly, what's happening is protons are being pumped as a result of the movement of those electrons. Now we generate a system that's like this, and all that's happening is ATP is being made, NADP is not being required. Yes? It doesn't have a name. You can call it cytochrome F if you want to. Okay. I won't ask you that.
So the electrons just like go through it and then it pumps the protons into the thylakoid.
It pumps the protons into the thylakoid. That's right.
But it's not like,
This thing, you can call it cytochrome F if you want to, but it's actually a little bit more complicated than that. But for our purposes, if you want to call it cytochrome F, I'm not going to ask you that name. Don't worry about that. Okay. If you know it as the proton pumping complex, I'll be happy. How about that? Yes?
Where are these electrons coming from?
The electrons are coming from right here.
From which molecule do they take it from? Because in the original one, it was taken from water.
It's taking it from this photosystem 1. Okay. So photosystem 1, it's kind of complicated. So I'm not showing you the source of electrons here, so don't worry too much about that. But the important thing is this is a source of electrons to drive this whole process. And the upshot is we're making ATP, or the plant's making ATP, without needing NADP. Okay? Very important. Now, this means that ultimately, yes, ATP will be in high concentration, the cell will run out of ADP, and everything will stop because there's no more capacity for the cell. It's got all the ATP at once, it's got all the NADPH at once, and it simply waits for those to be used. But cyclic photophosphorylation is a way of keeping ATP synthesis going as the NADP concentrations fall. Okay, that's enough for today. I have a song I promised yesterday that we'll sing today about cholesterol that is one of my favorites. I hope that you enjoy it. It's to a tune you've already heard twice and sung twice, so I hope you'll join me in singing this one. Some things that you can build with acetyl-CoAs are joined together partly thanks to thiolase. They come together, 1, 2, 3, 6 carbons known as HMG, and you're on your way to mega cholesterol. To synthesize a mevalonate in the cell requires reducing HMG-CoA as well. The enzyme is a reductase controlled in allosteric ways when the cell's impelled to make a cholesterol. The mevalonate made in metabolic schemes gets decarboxylated down to isoprenes. They're linked together willy-nilly to build a pp-geranyl in the cell's routines to make a cholesterol. A single step like farnesils, but that's not all. The squalene rearranges to lanosterol. From that, there's 19 steps to go before the sterols apropos, which you must recall to make a cholesterol. The regulation of the scheme's complex in ways inhibited by feedback of the reductase. And statins mimic, so they say, the look of HMG-CoA. So we sing their praise and not make cholesterol. Okay, so I'll be back to my office if you want to come back and talk to me about your exam.